Hello, everyone. Good morning or good afternoon, depending what time are you looking at me at this presentation of mine. And welcome to the Autism One 2020 Virtual Summit. I wish I was with you in Chicago, attending the meeting as I have done for the past 7 years, but unfortunately, because of this coronavirus emergency, we are recording from my office. The title of my presentation is a novel strategy combining the benefits of an original vegetal GC-MAF macrophage activating factor with the benefits of hemp fermentation and the presence of a unique array of probiotics and phages. Before I start, I wish to acknowledge all the efforts by Mrs. Terry Oranga, who has been organizing Autism One for many years, Mr. Ed Oranga, who has organized this virtual summit, and the technical staff for having organized this virtual summit in these difficult times. Of course, I wish to thank all of you who will have the patience to listen to this presentation. And I wish to thank Dr. Antonucci for his contribution to our research, and a special thank goes to Dr. Dietrich Klinghardt, Dr. Christine Schaffner, and all the staff at the Sophia Health Institute for their contribution to the development of the Ruggiero-Klinghardt protocol, in particular, the protocol tailored for autism. Before we start, also it is important for me to share this advisory. And as you know, no information in this talk is presented by myself as medical advice. And of course, I recommend to follow standard of care for autism and any other pathology, as well as rules and regulations established by health authorities of each country. I don't know which country are you in while looking at my talk, but I recommend don't go into troubles and always be sure to follow rules and regulations established by the health authorities of your country. I also would like to disclose my conflict of interest because I am the inventor of a number of formulas, supplements, and I develop the approaches and the products that will be described in this talk. In order to avoid any bias, however, the results presented here today were obtained by independent laboratories in Germany and in Belgium. So these are independent studies done by laboratories that have nothing to do with us. So there is no bias on the results. I wish to begin my presentation remembering my friend and colleague, Dr. Jeff Bradstreet, with whom we published 2 seminal scientific articles on autism and whose work on GC-MAF inspired the research that led to the results I shall show today. In the next slide, the first article we wrote together that describes the role of ultrasonography in the diagnosis of the lesions observed by Dr. Bradstreet in the brains of autistic children. The impact of this article, published in Frontiers in Human Neuroscience, is very high. It has more views than 94% of all articles published in Frontiers, and it is in the top 5% of all research outputs scored by Altmetric. And by the way, this article can be freely downloaded. This is the article I'm talking about, and I think you can read for yourself the conclusions. TUS, that stands for transcranial ultrasonography, may be a useful screening technique for children at potential risk for autism spectrum diseases, which, if confirmed with repeated studies and high-resolution MRI, provides a rapid, non-invasive qualification of extra-axial fluid, that is accumulation of fluid in the brain, and cortical lesions, as we described in the brains of autistic children. Now, ultrasonography for the diagnosis and treatment of autism is now in place in clinics all over the world. And I would like here to mention the excellent work that Dr. Antonucci is performing with ultrasonography in his clinic. The following image in the next slide comes from Dr. Antonucci's personal archive, and it shows the brain of an autistic child whose symptoms are improving in association with a protocol of manual lymphatic drainage that we described in 2019. The protocol is aimed at removing the lymphatic blockade that leads to accumulation of metabolites and toxicants in the brain. Also, this paper is freely available to the public, and you can read the protocol at this address, at this document identification number. The title of the paper is Clinical Experience of Integrative Autism Treatment with Manual Lymphatic Drainage, and the entire protocol, developed together with Dr. Antonucci and Dr. Klinghardt, Dr. Scheffner, is available here. Now, this is a typical image of the brain of an autistic child, and I will not go into the details because it could become too boring. But in any case, this is an area called the subarachnoid space where a fluid tends to accumulate. In this case, the accumulation of fluid here is normal. As I said, as actually Dr. Antonucci told me, this child is on the way to recover. However, there are still some lesions here in the meninges. They look like a binocular. I know these things might be difficult to appreciate unless you are a radiologist, but this is just to give you an example of how this technique is now being used. In addition, ultrasonography for diagnosis and treatment is now incorporated in the Ruggiero-Klinghardt protocol tailored for autism. That is a joint research collaboration with Drs. Klinghardt and Antonucci. In the following slide, the scientific article that describes the protocol step by step. It's very pedantic, but if you follow the protocol step by step, you can appreciate the complexity, at the same time, the practicality of the protocol. Also, this article can be downloaded for free. It has been published in the American Journal of Immunology, and it has many, many parts. It's long, it's a step by step. You can take a look and read it at your will. Now, let's move on, and let me show you in the next slide the other article published with Dr. Bradstreet. This article describes the role of the brain lymphatic system, or glymphatic system if you prefer, in the onset and development of autism. The impact of this article, published in Frontiers in Neuroscience, is even higher than the previous one. It has had more views than 99% of all articles published in Frontiers. So we may say that our scientific contribution to autism research is in the top 1%. Also, this article can be freely downloaded. This is the title of the article, Commentary, Structural and Functional Features of Central Nervous System Lymphatic Vessels. It was published in December 2015, regrettably after the passing of Dr. Bradstreet. Dr. Bradstreet was so kind to mention our collaborative research in his talk at Autism One in 2015, his last talk at Autism One. When he described his own therapeutic protocol, he was so kind to say that his protocol was inspired by myself as his good friend, colleague, and mentor. As he said, what I'm going to refer to as the Bradstreet Essence protocol. So I was inspired by my good friend and colleague, mentor, Marco Ruggiero. And then he goes on, you can take a look at his wonderful presentation that, as far as I know, is still available on YouTube. Now, one of the several talks that Dr. Bradstreet gave at Autism One in 2015 was entitled, How Close Are We to an Autism Cure? In his talk, Dr. Bradstreet talked about a number of strategies, and among other topics, he focused on 2 points, GC-MAF and HEMP. GC-MAF stands for GC protein-derived macrophage activating factor. And he said GC-MAF has been one of the most powerful tools that I have ever used in autism. And actually, his words were substantiated by publications, because 3 years earlier, in 2012, Dr. Bradstreet and colleagues published this paper in Autism Insights, describing the effects of GC-MAF on autism symptoms and on nagalase. Essentially, what they described is that as the symptoms improved, nagalase, an enzyme that is in serum, it is elevated in autism and in other conditions and may be associated with immune system dysfunction, decreased. So essentially they saw that with administration of GCMF, they observed improvement of the symptoms, at the same time a decrease of nagalase that was elevated in autistic children. This was published in 2012. 1 year later, Dr. Thayer, one of the colleagues of Dr. Bradstreet, who had co-authored the previous paper, published together with colleagues from the University of Florence, from Cambridge, United Kingdom, a paper in the American Journal of Immunology, where they reported, among other effects, also positive effects of GC-MAF administration in autism. Now, since 2011, so before all of this, we had developed a novel approach to produce natural GCMF by fermenting milk and colostrum at first, and then we went on to non-dairy products. Our goal was, and still is, to exploit the synergism between GC-MAF that rebalances and stimulates the immune system and the reconstitution of the healthy gut microbiome that, as we know, is a key element in autism, as evidenced by the many articles on this topic. You can go to PubMed and look for autism and gut microbiome or microbiota, and you will end up with a number of articles. This paper, published in 2011, is our first contribution to the field of GCMF, natural GCMF produced by fermentation of milk and colostrum. In a very recent scientific article, so moving almost 10 years forward, we demonstrate that in our fermented product, there is 100-fold more GC-MAF activity against nagalase than in purified GCMF. So our GCMF activity is 100-fold more potent than the purified GC-MAF that was used in the past. Now, in order to avoid any bias, the experiments that I will show you were performed by an independent laboratory, one of the best in the world for the study of nagalase and GCMF, the RED Laboratories from Belgium. And I take this opportunity to thank Dr. Tanya Mihatovic for all her help and powerful insight. I apologize. The experiments were performed in vitro, so to avoid any confounding variables. So we are not looking here at experiments on a complex organism or even on a cell culture. In the test tube, there were only human nagalase and our product, or human nagalase and purified GC-MAF as a control. The following slides show the scientific article and the results. This is the paper that was published in the American Journal of Immunology, complex and long title, together with Dr. Michael Carter, where we demonstrate the effectiveness of our approach on nagalase and on other markers of systemic inflammation. This is the slide that shows how the PMF, or product of microbial fermentation, diluted 100-fold, so 100-fold more diluted, was still always more effective than purified GCMF, both at 0 time and up to 3 days later. Actually, we have results that are published in that paper demonstrating that up to 120 hours after the beginning of the reaction, still our product was 100-fold more efficient than GC-MAF in binding nagalase. And as you remember, nagalase was the key element, the key enzyme that Dr. Bradstreet had observed as a key element associated with improvement of symptoms in children with autism. This slide reports the same results as before with the different graphics, just to show you how in yellow our product of microbial fermentation is always 100-fold, more than 100-fold more efficient than GCMF in binding human nagalase. This 100-fold higher activity of our product in comparison with purified GCMF can be explained taking into consideration 2 elements. First, naturally formed GCMF in our product is not alone. It is associated with vitamin D3, fatty acids, and glycosaminoglycans such as chondroitin sulfate. All these are normal constituents of milk and colostrum. And actually, since 2013, in this paper published in Nutrients, you can find it on PubMed, we proposed a molecular model describing how non-covalent association of all these constituents in a multimolecular complex significantly enhanced the biological activity of GCMF. So it came as no surprise. We were particularly pleased to see that quite recently, at the end of 2019, researchers from Germany and Austria demonstrated, independently of course of us and indirectly, that this concept of ours was correct, because they confirmed that association with vitamin D3 significantly increased the immune stimulating activity of GCMF. You can take a look at this paper. It is also this one retrievable from PubMed. The second element that explains why our product is so much more powerful than purified GCMF is the presence of phages or bacteriophages. Phages are in our product. We have described them in full detail in a couple of papers. This one on the right, published in bioRxiv, and one that will be shown in the next slide, published in the Journal of Neurology and Stroke. Now, these phages, they synthesize proteins that have activities superimposable to that of GCMF. And these proteins complement the effect or add up to the effects of the naturally formed GCMF that is present in our product. Now, we have recently characterized in detail the composition in phages. You could call the phages the friendly viruses, and in this time and age, it is important to say that these viruses are very friendly to us because they are those that kill the bad bugs, if you want to call them so. So you can read about the bacteriophages or phages in our product in this paper, and also in this one that quite recently was accepted for publication in the Journal of Neurology and Stroke. It's a long list of phages with a long list of biological activities that in some cases have been recognized for more than 100 years. For example, just to go into the molecular detail, one of these phages that is called the Lactococcus phage UL36 synthesizes a protein, and this protein is homologous to a human protein that is called RAD52. The viral protein is called SAK, and the human protein homologous is called RAD52. Now, interestingly, as this study, original study of ours demonstrates, this protein shows a significant similarity with the active site of GCMF. On the top line, you have the active site of GCMF. These letters, they indicate the amino acids. On the lower line, you have the protein synthesized by the phage, the homolog, the human protein. And I don't know how familiar are you with the similarities in amino acids and proteins, but let me tell you that all these little symbols indicate that they are very, very similar. As I said, alignment of the two proteins has showed a striking similarity between the active site of GCMF, that is the sequence of these amino acids, TPTLAK, looks like when you go to the optometrist, and the sequence, you can read for yourself, of the protein of the phage. In other words, in our product, we have GCMF coming from fermentation, as I said before, and as we demonstrated since 2011, plus we have GCMF-like proteins that are produced by phages. Now, this leads to some interesting considerations, because production of these GCMF-like proteins by phages does not come as a surprise. It is well known that phages modulate the immune system, activate macrophages, help fighting infection, and exert anti-inflammatory actions. These words in italics are from an article by scientists from Belgium, California, Poland, and Australia, describing the profound positive effects of phages on the human immune system, as they are centered on macrophages, as the following figure from their article shows. They say phages also impact immunity directly in ways that are typically anti-inflammatory. And you will see in a short while why this is important. Phages can modulate innate immunity via phagocytosis, that is what macrophages do. and also impact adaptive immunity via effects on antibody production and effector polarization. So these slides comes from this article. As you see that macrophages are at the center of the effects of phages on the immune system. And as we know, GC-MAF stimulates macrophages. But also interesting is to read the caption to the figure of this paper that says, independent of the route of administration, phages can enter the bloodstream and tissues and encounter immune cells in the blood. So this means that if you eat phages with a product that contains phages, they reach the immune system. And again, this is not us saying these are the esteemed authors of this very interesting article published, is not a coincidence, on a journal called Viruses. Now, why do I want to stress that phages impact immunity directly in ways that are typically anti-inflammatory? Because in these days, you probably have read many, many times about cytokine storms. Cytokine storms in these days of the coronavirus are mentioned as one of the main causes of complications and death due to the viral infection. Actually, cytokine storms are not peculiar of this coronavirus or any other virus. They occur in a number of conditions. I found these in the US government publication about cancer that says that the cytokine storm is a severe immune reaction in which the body releases too many cytokines into the blood too quickly. A cytokine storm can occur as a result of an infection, autoimmune condition, or other diseases. And of course, cancer in particular, it may occur after treatment with some types of immunotherapy. And signs and symptoms are those that you can read. So cytokine storms, they may occur in a number of conditions, and essentially they represent an exaggerated response of the immune system. And cytokine storms also have been implicated to some extent in the pathogenesis of autism. Here I found a couple of papers, but you can find many others. Just look for cytokine storms and autism. And these are two papers that describe how cytokine storms may be implicated in autism. What about phages? What do phages do? Can they prevent cytokine storms? Apparently they can. You can take a look at these papers published in Frontiers in Immunology in December 2017, where the authors, they explain the potential of phage therapy, in this case, in severe infections of the blood that are called sepsis. And they describe how phages, because of their anti-inflammatory immune stimulating activities, can be helpful. If you want to know more about our approach to viruses, I take the liberty to highlight this very recent article of ours, published with the researchers from Thailand, describing our approach in viral infections. But let me move on to the second topic that I want to cover today, a topic that was also described in full detail by Dr. Bradstreet in his last talk at Autism One, hemp. Actually, the work that Dr. Bradstreet performed on hemp-derived compounds was a collaboration with some colleagues of ours, of mine, Dr. Antonucci, Dr. Siniscalco, and colleagues from universities from Italy. Now, hemp. We have developed an approach based on fermentation of hemp proteins in order to combine the effects of reconstituting the healthy human core microbiome that we have seen is a key element in autism approach, together with the effects of fermented hemp proteins. Now, I don't know how familiar are you with hemp proteins. Nowadays, everybody is familiar with the CBD, maybe THC, so all these psychoactive substances coming from several different parts of the hemp plant. Hemp proteins have nothing to do with psychoactive substances, and actually in the hemp seeds, there are two types of proteins that are called globular, because they have like a roundish shape. One of them is called edestin, and it is about 60 to 80% of all hemp proteins, and the remaining is albumin. Albumin, just like the albumin you have in the egg white or in our blood. According to Wikipedia, so I'm not making up anything, edestin has the unique ability to stimulate the manufacturing process of antibodies against invasive agents. So it is immune stimulating. Edestin is similar to serum globulin, those globulins that are in blood plasma, just like this GC globulin that is the precursor of GC-MAF. And the biologically active protein of edestin, which is something that makes a little sense in biochemistry, the way it is written, but in any case, I just copied what's written in Wikipedia. It is metabolized in the human body and it is capable of biosynthesizing hormones, which of course regulate all body processes, hemoglobin, which transports oxygen, carbon dioxide, and nitric oxide, and it is essential for our life, enzymes that catalyze and control biochemical reactions, and most important, from our immunological perspective, antibodies, immunoglobulins, which fend off inviting bacteria, viruses, and other pathogens, as well as toxins or antigens as they enter the body. Now, edestin is so similar to human blood proteins that there is even a patent application proposing its use as a substitute for human plasma. And the abstract of this US patent application reads, the agent for substitution of the blood plasma is made up of the vegetal protein substance edestin, advantageously a product of the hemp seeds. So, in other words, one of hemp seed proteins, edestin, is very, very similar to human globulins, so similar that it could, at least in theory, even be used as a blood substitute. Now, such a similarity to human blood proteins led us to investigate and compare edestin with the GC protein that is also known as the vitamin D binding protein, that is a globular protein of human blood that is the precursor of GC-MF. Now, this figure on the right, that again is the result of alignment of two proteins, the edestin from hemp and the GC protein, human GC protein, shows striking similarity, high degree of similarity between GC protein, that again is the precursor of GC-MAF, and edestin. Now, if you want to read all the details, please go and see our article that was published quite recently. Again, also this article, like most articles of mine in recent times, is freely downloadable, and it can be found in Medical Research Innovation 2020. And over there, you will read all the details, how we were able to produce vegetal GC-MAF from edestin, from fermentation of edestin. We were able, because we were able to identify the regions of edestin, the region that is homologous to the active site of GC-MF that we had identified many years earlier, and we used this information in our approach. Now, again, if you want to play with amino acid sequences, this is the same active site of GC-MF that I had shown before, and here in the lower line, there is no viral protein as before, but there is the sequence of edestin. And also these little signs here indicate the very high similarity between the active site of GC-MAF and the fermented protein edestin. In short, as I said, we were able to produce hemp-derived vegetal GC-MAF in a probiotic that also contains all the microbial strains mentioned before that we know are essential in reconstituting the healthy gut microbiome. And as I said before, you can go and take a look at this paper of ours just published. Efficacy of our fermented egg product was assayed in vitro by a major laboratory in Germany. As I said, we asked independent laboratories to perform analysis on our products, so to be sure that there was no bias or preconception on our part. And this laboratory demonstrated that our fermented egg product had as the biological activity of GC-MAF. All this is written in the paper. Now, something that also is worth mentioning is that in addition to fermented edestin or edestin, our product also contains fermented hemp albumin. Albumin, just like edestin, is a protein with known healthy properties, as briefly reported in the following slide. You know, you could write encyclopedias on albumin, but just to make it short, it is the major source of extracellular reduced sulfhydryl groups, which act as potent scavengers of reactive oxygen species. So it is a very powerful endogenous, if it is our albumin, or exogenous, if it is albumin coming from hemp fermentation, antioxidant, because it takes away the reactive oxygen species. And the antioxidant function of albumin resides also in the capability to bind and eliminate several metal ions, including copper, cobalt, nickel, zinc, and iron. And I think you are all familiar with the role of metal toxicants, improperly called heavy metals, in the pathogenesis of autism and other neurological conditions. As a result, albumin represents the main circulating antioxidant system in the body. Again, if it is our albumin, but also you can assume albumin by eating egg white, if you like, or fermented hemp proteins, as in our case, and that albumin has all these antioxidant actions that I've just shown. But more interestingly, this is a little bit of biochemistry. I know it's boring, but is just to give you all the elements to evaluate our research. Fermentation of albumin leads to the natural production. Here there is nothing artificial, nothing chemical. All these reactions, they occur as part of the process of fermentation, which is the most ancient way humans had to process food. So natural production of albumin anhydrides. So what are these things? Well, essentially, during fermentation, some molecules like a succinic acid that is formed during fermentation bind to albumin. And this leads to some very interesting properties, because albumin anhydrides have powerful antiviral effects against a variety of viruses. This paper was published by Chinese researchers in the Journal of the American Society for Microbiology, and they write, in our previous studies, we have demonstrated that several kinds of viruses, including HIV, human papillomavirus, and the respiratory syncytial virus, can be inhibited at the viral entry step by anhydride modified proteins. So in other words, these anhydride modified proteins, and albumin in particular, because that's the title of their paper, albumin, potently block the entry of a number of viruses from Ebola to HIV, to HPV, and so on. Interestingly, this peculiarity is shared also by a bovine protein, the beta-lactoglobulin, that is in our other approach, the one fermenting milk and colostrum. So different ways to achieve the same goal. Now, you probably know that chronic viral infections have been hypothesized in autism, and also this is demonstrated by a number of scientific articles that are published on this topic and are retrievable in PubMed. Here I listed a few of them, from the Zika virus to the rubella virus, to human endogenous retrovirus in Albanian children, and so on. So you can read a number of papers at least associating viral infections with some of the symptoms of autism. This was the last slide, and these are the conclusions. The conclusions, to make it short, are that independent laboratory results have demonstrated that our fermented products have 100-fold more GCMF activity against the human nagalase when compared with purified GCMF. Our product with fermented hemp proteins exploits the properties of idestin and albumin, two proteins with known health-promoting immune-supporting activities. And we also demonstrated for the very first time that idestin shows a striking sequence similarities with GCMF, thus playing the role of a vegetal GCMF. I thank you again for your time and attention, and I hope to see you in person as soon as possible. Thank you again, and bye-bye.